Hello, I'm Patty Paulson, and I do the science education at Bethel University. Inquiry-based learning has kind of been a buzzword too much lately. But in reality it's the way we learn from the time of birth. I watch our grandchildren, and even as little ones, everything is, what's this? Why? What color? When? And then at a certain point, they get about intermediate, middle school, and they learn it's dumb to ask questions. And they get afraid of asking questions for fear they might appear dumb. But you look at even the smallest infant. They put things in their mouth, they taste them, they manipulate it, they inquire about it in their own way, in ways that are interesting to them. It's how the brain actually forms patterns by meaningful links to what they already know. We know from the brain research that questions are what wakes up the brain. The I wonder kind of question. If we give an answer, I love to give the example when I'm out in a nature trail. And someone says, what's that tree? I said, it was a buckthorn. They're never going to remember again what my answer was because there's no meaningful connection to it. But the brain is satiated, it's satisfied, and we walk on. And I'm seen as the sage. I could have given the wrong answer. It doesn't really matter because they won't remember it and there's no connection. If, on the other hand, if I have them go out and actually look at the tree, look at the thorns, look at the berries, look at the leaf patterns, look at, the bark. The next time they see it, they'll not only know it's an invasive species, but they'll remember the sight, the touch, the sharp thorns, all the things about it, and they'll remember for themselves. We kind of give kids the impression there's one right answer to everything. And if you know it, you're smart, and if you don't, you're dumb. And so kids are real afraid of taking risks that way. Inquiry-based learning allows us to actually look at things that have multiple solutions, solve problems, and look at things from different angles. The old saying is, if you're just going to cover something in class, you might as well dig a hole and cover it, because it's never going to be remembered. And we hear teachers all the time say, well, I covered it, they didn't learn it. Well, that's because it was all about the teacher, not about the student. Inquiry involves the students. If you look back to Socrates, you look back to Dewey, you look back to Vygotsky, they all were active, engaged, learner-based teachers. There's a dance that occurs. Schoen says that there's a fine line between when we tell kids what we need to have them know. So there's a certain part of inquiry that says we need to give them some background to help them actually do the inquiry. But if we tell them too soon, they won't be ready. And if we tell them too late, they won't be interested. So it's that interplay of formative assessment we have to do with students all the time to say, how do you know that? Are you ready for this? Do you understand? If we really listen to the students, we gain insights into what they're thinking. We think about the brain, that little 3-pound thing, or maybe a little more. Every one of those brains out in the classroom is constructing their own understanding of what's going on in class based on what their prior experiences are. It's pretty daunting for a teacher to think about that because we're thinking about what we're presenting, what we're doing. But in reality, they're grasping it and processing it and constructing their own understanding of it. And if we don't take time for that, to listen to that metacognitive process, which occurs very naturally through inquiry, we don't know where they are. And we move on often before they're ready. We assess too soon, we move to the next topic, and all of a sudden we're moving on and nobody's following. So to actually have that active-based learning looks a little different in the different disciplines. Social studies, literature tend to be more primary, secondary source inquiries. Math and science involve more actual data collection kind of things. I've been working with a lot of the people in the science inquiry, obviously, because that's what I do. And with science inquiry, we're looking at authentic experiences to actually try to put them in the world of a real scientist at a developmentally appropriate stage, based on their own background and their own understandings. But when they can form their own questions and inquire about them and dig into them and come to their own conclusions. And there's not one right answer. We find it's a much richer learning process. One of the misconceptions on inquiry is anytime you do hands-on, you're doing inquiry. That's not true. If they're just working, what's the saying, worksheets don't build dendrites. If they're just moving through, what do you want? What should I put in this? How long should it be? Is this okay? Then there's no inquiry being involved. They're just trying to get into our heads and reproduce what we want them to reproduce instead of taking ownership. The more we can provide an atmosphere that allows students to ask questions and wonder and want to go further on things, the more productive the inquiry. Once again, if the brain's satisfied, it shuts down. So if we're not at the correct level of difficulty, the brain shuts down. It goes into survival mode. It just, what do you need? What do you need? What am I supposed to do? What's going to be on the test? Instead of actually allowing them to ask questions, to wonder, to try to understand. And so if we ask the questions, it may be at the wrong level of difficulty, it may be something they're not interested in, and we could lose them right away. So knowing your students well and creating that atmosphere where they feel free to ask and discuss things is so much richer. The other thing about math and science, there has to be evidence-based. And they have to tie that evidence to actual concepts so that they come formulating their own conclusions and defending them based on the evidence. Instead of just doing a math problem for the sake of a math problem, it can be beautifully incorporated into a science graphing analysis kind of program and actually have them come up with their own conclusions. Inquiry involves higher level thinking. Certainly there needs to be knowledge comprehension base, but it's the analysis, application, synthesis, evaluation pieces that really make inquiry rich. Well, the other thing about inquiry-based learning, I think, is that it often is misconstrued in terms of, well, if you're only doing cookbook activities, then that's not inquiry. We have to think very carefully about the purpose of our instruction and what we want them to be able to do. There's 4 levels of inquiry. There's the old confirmation, where I know what I want them to know, and they know where we're going. The outcome is very clear, but I want them to see what I want them to see. I don't want to have any risk that they're going to get misconceptions on it. So if I'm doing the potato borer lab, I want them to see osmosis as it's occurring. I don't want them to have something go wrong and not have it work. So in those instances, a confirmation kind of lab is very appropriate. It's much better than just having me tell them what osmosis is and memorize the definition. I haven't had a student ever that hasn't heard the word, but they can't tell me what it is. So if I actually have them seeing it and observing it, that's a very appropriate use for a confirmation type lab. If I've got students who are inexperienced in inquiry, it's much more comfortable to scaffold that instruction, to have them start out with more confirmation and gradually open it up. The next stage is the course structure, where we present the question that we're looking at. A classic I do is, what are the characteristics of mealworms? Obviously a segue into insects in general. But I have all kinds of materials out. And as you know, I teach an elementary majors science course. And so it's something they can take to their own classroom. They formulate their own hypothesis. They do their own experimental design. So one might be looking at light, dark, somebody else might be looking at hot, cold, somebody else might be looking at sweet, sour kind of things. They come up with their own, and together we come back as a community of scientists and synthesize what we found out. And often they'll have 2 groups with the same question, different results, and it ends into a very rich discussion. But it's much more structured. The topic is there. They have a wider range in what they can do with the topic, but it is structured. Guided is where I let them come up with their own questions a little bit more. One of my favorite examples of that is what I do with the methods courses, where I try to model children's engineering that they can take. And they have to actually create things as an engineer would. The new science standards, K-12, have engineering strand completely through it. And many of our students haven't had experience with that. So I'm trying to build that in as much as possible. So they might have to make the best top in the world. Obviously, we start out with having to play with all kinds of tops. Many of them have never played with a top before. That's something us more vintage people have played with. And so then they have to create that whole thing talking about center of mass, balance, centripetal force, as they move through it, where's the force applied, and then they decide what materials are best. We do the same thing with puffmobiles, roller coasters, those kind of things. And so it's guided in the sense that I'm the guide on the side, asking them questions, giving them some things to think about, some things to maybe hook to prior learning, whether it's referring to the ice skater in the Olympics or whatever, and really think about that and how what happens when they put their arms out, pull their arms back together, those kind of things, and then let them actually move through that inquiry themselves and present it back. It's not a competition. That's a really important piece. Example of that I know in the K-12 schools is they do a bridge unit. Well, the ones that are doing the elementary engineering, when the 35W bridge collapsed, they were home talking about struts and bridge plates and weight and where it was being distributed, because they built bridges. They tested them. They tested out materials. They didn't just memorize the difference between a, truss bridge or others. They actually had tested different types. They talked about triangles. They understood those pieces. And much more rich, exciting kind of discussion going on. Open inquiry, I'm not totally comfortable with. That just sort of says, what would you like to learn this year in school? And it really requires, I call it the teacher on roller skates kind of thing. One of the things I think that's really changed with my teaching in this whole thing is that I teach from the back of the classroom so much, because I have the airliner slate, I've got the PowerPoint on a smart board, I've got the document camera, and I've got the students up and involved in doing things themselves, so that I'm not the sage on the stage. I'm not there to show them how smart I am. I've already learned it, I know it, so what? The point is for them to own it and become a part of it and have them internalize it themselves. And so they are making the presentations and showing what's going on. And if they haven't been trained that way, if they've been trained with worksheets, if they've been trained there's one right answer, if they've been trained here's the study guide to memorize everything, they're not very comfortable with it. And it takes a while of trust building. But usually in the end they come around to really appreciate at least the depth of understanding they've been developed. What do they say? The American curriculum is an inch deep and a mile wide. Sad but true. The things I'm teaching are things they should have learned often before, but all they've done is memorize. And of course, since I deal with teachers, they have to have an understanding in order to teach it, so it's an important fact. I think we've all been through the sit and get lectures. Sort of like sponges, we sit and get. And in reality, what is that motto? Teach me and I remember. Tell me I forget, teach me and I remember, and involve me and I understand. It's an old adage, but it's very true. We find that if we only present information, the brain has about 18 seconds to decide what to do with new information. You're constantly being bombarded. One of the things I do with my students is say, think about your small toe. Well, they weren't even thinking about it before, and now suddenly it's come to the front of the frontal cortex, right? And or frontal lobe. And we have to hone out things all the time. And so even though today's students are quite good at multitasking, the reality is they may or may not be tuned into what you're doing at the time. And so that idea of actually giving them a chance to rehearse it, to put it in the short-term memory, and then in the long-term memory. What if we actually see something, hear it, and talk about it with another person, and actually manipulate it, where our memories goes from 10% up to 95%. We know that. So the more we can actually have them do the inquiry process to talk to each other, those kind of things, the better. We want to create productive future citizens. That's what we're here for. We're not here to just have them tell about what a wonderful experience they had in college and not remember anything. We want them to be productive citizens. And I continually get discouraged with the level of discussion that's occurring on the national and world level. We look for little sound bites. It sounds like a good idea. Elections are done with sound bites. People can discuss climate change as if it was no different than a change of shoes. They don't understand all the various impacts of different forces and all the things that enter into it before we say is there or isn't there, and what's causing it. One of the things with science we're pretty comfortable with saying is we don't know, but we want to find out. And I think that's often been misrepresented in schools, in the sense that there is one right answer and scientists know all the answers. No, good scientists know we never prove anything. We're always open to new ideas. I think about, in my lifetime, how things have changed with new technologies, that the nanotechnology that has never been available, the undersea exploration, the deep looking into rainforest ecology, those kind of things. They weren't even part of what we could do in the past, and now we can. We can get places we hadn't been able to get before. We can visualize things in ways we've never been able to before. And if we're just going to give students facts, they can sit there with their little phone and Google the answer and know it right away. That's not what we're doing. So I really want to have them question, wonder, dig, find out more, and really care to make it their own understanding, not just what somebody says they should believe. And if we can do that, obviously we work with the teachers of the future. That's what we want their students to also do. We don't want them to actually just put in their time getting ready for a test. The real test is life. Well, I alluded to it a little bit before in the sense that we want them to become lifelong learners. And the way they become lifelong learners is to be an active participant in the process. That they care about their learning. They're not just caring about what grade am I going to get. They want to see what they understand, how they can process it, how they can come up with their own thinking on it. So it's theirs. It's not mine that I've handed in a nice little package for them to take away. And if we could create that, Project 2061 in science is a good example. The idea is we want to be tops in math and science by the year Halley's Comet comes back again. We're not moving in the right direction right now. And the only way that can happen for there to be scientifically literate citizens is to take problems, issues, concerns, and really work on it. At Obama's first State of the Union address, one of the people sitting with Michelle will be a high school girl who has been accepted to Yale, and the reason she has been chosen for this, again part of the STEM initiative, is because she's working out on bioluminescence, biology, things that light up in a very simple way, markers in water to determine purity of water. And it's a high school girl. She didn't figure that out by sitting and memorizing. And I think that's really important to look at. Kids can do a lot more than we think they can do. Our students can do a lot more than we think they can do. We need to really ask them to try that and to really go to that. Reality is, inquiry learning probably takes more time. I can stand up there and lecture for 45 minutes, deliver the same amount of information. And sometimes that's appropriate. Sometimes it's not worth the time, for example, if we're learning, I don't know, parts of an insect. There's no point in having them do that inquiry learning. Maybe at first grade, not at college. If they've forgotten, give them the labels, give them the terminology, give them the equipment they need to work with, and then take it to the next level for the inquiry learning. So sometimes, and sometimes the inquiry learning needs to be a little bit more confirmation type, like I mentioned. Depends on purposes. I think one thing we need to be very transparent about with ourselves and with our students is what are our outcomes? What do we want them to do? If we want them to simply know about something, then lecture. They know about it. If we want them to comprehend, then have them do a little practice of it. Worksheets work real well on that. But if we want them to analyze, synthesize, evaluate, and apply it, then we get into inquiry-based learning. And the reality is we know that we will not have long-term retention with simply knowledge level things. We can state a lot of facts, but if we can't do anything with it, it doesn't count for anything. One of the things that we need to be very careful of is to uncover what the student's thinking is. There's where we determine, do we need a little bit more practice, and is it worth the time? If I'm having them do performance assessments, developing rubrics and checklists that are truly quality checklists, that takes a lot of time and a lot of practice. To me, it's worth it. If I want them to memorize the theorists, they can do that on their own. They don't need me for that. But they do need a coach to help with language, to help with understanding what understanding should look like at different stages, and to understand that whole realm. Excuse me. If they need just background, then we just give them the background. And we have to really be intentional about our purposes. If the educational goal is for students to understand things deeply, then they need to know how we come to understand what we're understanding. And that's where inquiry is most effective. In the piece you're about to see, we're about 1/3 into the course. And we've already looked at cells and characteristics of cells. And now we're looking at, we've been through bacteria and fungus, so they've looked at prokaryotes. We're just starting into that kingdom of the protists, and they're looking at how these are more like eukaryotes and prokaryotes, and different characteristics of protists. All right, protist paradise. I got one? Would you turn it over to the back side of it, where you'll see a T-chart, okay? We're not going to do anything with it yet, but just that's the side you'll be looking at. The other you'll be working with your partner with. So, remember when we first started talking about cells, and we talked about how they first saw in a cork cell, right? Well, Leeuwenhoek was the first to see living organisms moving. And we talked about how we've been able to uncover lots of different things with disease and other things because we have greater technology. Well, imagine being able to see this for the first time, which is what he did, and he described it, little eels or worms lying all huddled up together and wriggly, and the whole water seemed to be alive with these animalcules, and cells filled with juices. Looking at exactly what you were just looking at a minute ago. Scientists don't prove anything, but we find substantiation for it. So what I'd like you to do with your partner for just a minute is to take a look again at those animalcules that you see floating around. And with your partner, talk about some claims you might make about what you're seeing and what you know already based on the text in our lecture today about these organisms and what evidence you see that would help you support that. A few minutes and write that down.
Someone who explained it in kindergarten, the kingdom.
Write down a claim you would make.
Chloroplasts, so they have photosynthesis, but then the big longer ones, would they be...
Oh, the smaller branches.
Hold on one sec, guys. These are smaller. Oh, look at it now. Bacteria evidence for that would be...
That's the upper right? Yeah, it is.
What do you notice about the things around it? Some rather large ones, you can really see the inside. Look carefully. Let me change focus again slightly so you see another layer of the water. One of your clues is how does it move? Is it 9-2 arrangement or is it simple whipping? See if you can observe that. Complexity like a socket ridge, right? Okay, good. Flagella? Remember the prokaryotes are real simple, they're just whipped. Okay. Everything else, eukaryotes, have that 9-2, so it's more like a socket ridge that moves like sperm would, so it can move and be much more flexible.
So it moves around more.
Yeah. As compared to just look fixed. Yeah. That's what flagella is, right? But remember, it can be all the way around, too, in a cilia. Much more. Well, what would be the difference? Let's look at it this way. What's the difference if you have like a fixed wrench that you go to take a bolt out? Fits right on that one, right? And you just kind of go back and forth, right? A socket wrench has a socket on the inside, so it can move in different directions more easily. Exactly. So the eukaryotes have that 9-2 arrangement. There's 9 little attachment points, around the flagella, so that it can move much more complex way than just whipping back and forth. Make sense? Watch it up there. See if you can find one.
Like the big ones that are just like.
Yep. And some of the more slow-moving ones. See that one coming across at the top?
Yeah. Can you kind of see it at the end?
And if we change the focus slightly, it's right under the surface tension, so it's not really good focus. When you get your own scope, you'll see it. But you see that going much more silly kind of movement? Claim is what you observe that, and you think may be true. Lowenhoek said they're animals. Like my granddaughter says, amnos, right? Is there anything to support that? Would he have any basis for that? That would be your claim. I see some without evidence. I'll give you another minute to come up with evidence. Okay, write them down. Can scientists claim anything without evidence? Okay, let's give an example. Like, okay, so you've got the ecosystem built, right? Okay, so what evidence tells you that something's on the top level? Okay, does that make sense? Under the evidence column, yeah. Under the evidence column. Well, these are claims you're making. They're all different sizes and shapes. What's your evidence? Yeah, but basically looking at the lens, right? Some would fill up the whole field, others wouldn't, you know. Because if you just looked at that drop of water, would you see any of that? No. Okay, so part of it is the field of viewing that you've got. Okay, some you're thinking one flagella is moving back and forth, which would mean it is a prokaryote.
really big.
Would you
Like
skip
the bigger
over
one.
in this slide? So do you have evidence for this? You do have an evidence that it's a flagella as compared to multiple ones, maybe, all around the outside. Okay? Right? Take a risk. Dan, okay. Why? What's evidence would you have that it's an ecosystem? Mm-hmm. Mm-hmm. Variety. Keep going. What has to happen in an ecosystem?
Feel that sustain nutrition.
Yeah, they kind of, there's something that eats something, right? Did you see any of that? Mm-hmm. Okay. You've got a whole ecosystem in a drop of water. Pretty interesting. Is it a healthy ecosystem? If I told you this is Lake Valentine, would you say Lake Valentine's healthy?
Probably good for bacteria.
Are you seeing bacteria? Oh, you are. What color is it stained? Is a gram-positive kind of showing up that's letting you see it? Remember we looked at bacteria, right? Okay. What do you remember when we looked at bacteria? What'd you do? Help me see it, right? Because it looked like measles on a slide, right?
Yeah.
1/10 the size. So are we seeing bacteria?
No.
It's there, but we're probably not seeing it because the magnification is only at 40. Did you see bacteria to 40 magnification? No. There's your evidence, okay? But does something else eat what's there here? Is there something else in the lake that is eating what's here? Yeah, there is. If we could go out under the ice and get an actual sample of the net, you would find all kinds of little mini crustaceans, little fairy shrimp that are eating all these kind of things, little copepods that are eating these kind of things. And then, of course, something else is eating those little fairy shrimps and daphnia, right? And something else is eating those. So if we wiped all these out, would the lake survive? No. This is a sign that we've got good oxygen, things are healthy. Okay? If not, the bacteria take over. Okay? So, whoa! What's your evidence that that's bigger than the other? Okay, give me another claim.
Different levels.
Different levels in the water. What's the evidence of that?
Because like one thing will kind of like spin over another thing and it won't, the thing underneath won't interfere with anything.
You're not seeing that in there. Almost like levels of a lake. All right. Do you notice when I change the focus, you're seeing different things? Yeah, that's another evidence that you've got not a flat slide. Okay? I'm asking you and your partner to grab a microscope, and then first of all, you have to go fishing to find your own ecosystem. So over here, there aren't quite enough for every pair to have their own little lake, okay? So we'll spread them out so that you can get different stations. But you need to get a fishing pole, and you need to get a depression slide. Why would it be called a depression slide? Yeah, it's got a little spot for you to put your lake in, okay? So you need to go and just get a little bit of the pond water and put it in the depression. You do not want it overflowing, okay? And again, remember you start with the highest, the largest field. Get a good spot where you're seeing lots of movement. And then you can probably take it up to the yellow magnification. Don't take it to the blue because you'll put the microscope slide. This, I don't want you doing inquiry on this. You'll get the microscope slide down in the water and then it's a mess to clean the lens, okay? So with your partner, make sure as you're doing the adjustments, you're not putting the microscope down into the water, okay? Just keep a good handle on that. Move it around. See how many things you can draw and describe. Okay, you've changed the magnification, obviously, because of those different levels that you've talked about. And you'll see many of some of them and just a few of others. If you think you found everything in there, you're more than welcome to come get a new sample. However, these are also God's creatures. Do not run them down the sink. Get the leg sample, the other
We'll just
disc.
use the microscope right there.
There's more in the cabinet than some of the table. If you find something really cool, we'll put it on the big one.
Yeah, because otherwise this will touch the thing.
Why wouldn't we just go down? She said it's
And when you get your sample, get around the little plant material. Good job. Yeah, watch it moving. Because sometimes, remember, they can have it all the way around, so you can look for that, or you can look for the single tail. Some of them you may see two coming out. Okay, so look carefully and see what you see. You have to watch them for a little bit. Can't see any? Let me see. All right, let's move it a little bit. When it's swimming and it flattens out. Okay, see what you see.
And what about like size? Just like
How do you
medium,
describe
or
it?
like
Yeah.
how would you describe size?
Yeah, you'll see some that are very, very tiny. You know, some little cobia, but just teeny. And then you'll see some of them that are great big. And so I think you can just go small, medium, large.
Okay.
Because you don't have to put in any kind of metric measurements on these.
I'd say medium. You got small.
Small, medium.
Alright.
Internal structure.
Now, it's a little thicker wide. You have to change the adjustment. A book that says Pond Life and a set of papers that says Common Water Organisms.
I gotta find that thing.
Okay, this is for your need to know. One other thing I'd like to notice. Yes. With me? Have you heard yourselves? Look at this! Man! Cool! Look how it's moving! What do you think fourth graders are going to do? Same thing, only they'll be less reserved about it, okay? So it is a fun lab. They like doing it.
What else? Oh, the one that changes shape. Maybe, okay, it grows and pollinate the cell sets and goes from here. Vaccines, chances that let's see what else is other organisms. I don't know. Because they're kind of gel-like. Yeah.
What did you notice differently when you were describing it as compared to when you had to put a name on it? So is the name you're putting on necessarily the right one? Probably not. Have we done enough in this class for you to know the exact right answer? No. The idea is to what? Appreciate the diversity within the ecosystem. Some are plants, some are more fungi, filament shaped to them, some are more animal-like, they have different sizes, different movements, okay? And I don't want you to go to fourth grade pounding in that they need to know the difference between a bleeding and a paramecium by sight. Okay? The important thing is to appreciate that even on a single drop of water, God's creation is immensely diverse. And the big factor that affects how it moves is that structure-function thing, right? That we've talked about and talked about. Okay? So if you didn't get all your boxes filled, that's okay. I'm looking for your drawing description more than anything. Okay? And so when you have finished, as much as you feel you can, then please return the organisms to the lake from whence they came. Okay? And return the microscopes. If there's any water on the lenses, take a Kim wipe and wipe it off. And Tuesday, I will see you then. as I think about what worked and what didn't work, I'm reminded that you never arrive at being good at an inquiry-based teacher. You're always trying to hone your skills to get better at it. As teachers, I think our natural tendency is to try to offer some sort of structure or control. And sometimes that needs to be less, and that really requires thinking about to make sure that it's less of us and more of them. I felt really pleased about this, using the science writing heuristic for this particular lesson where they were doing claims and evidence, because I think that really got them thinking carefully and deeply about what they were looking at. What I noticed in their lab reports when they came in, it was much more detailed observations, much more close connections to what we've been talking about with eukaryotes, whether it was what the flagella looked like, or movement, or coloration, or whatever. They were much more detailed. It also helped move them away from their traditional training of one right answer. This is a euglena, this is a paramecium, which wasn't my point necessarily. The course is based for elementary teachers to have a comfort level with the diversity within the protist kingdom, and to really look at, even within the single cell organisms, how structure, function, all the characteristics of life are very evident and very present. And it's a very fun lab. It's a very engaging lab. I've done it with children, and they, come here, come see. College kids aren't any different. They were just as engaged with it. So providing some structure, some focus to it, I like doing the claims and evidence piece of it, and to move them a little bit past the one right answer thing. I was recovering from a cold, so I didn't feel I was maybe as sharp on my feet to elicit answers from them as sometimes, but I think that's true of anyone. It requires a high energy level to do inquiry and to make sure that you're keeping them engaged, keeping them focused, keeping them on task. And so I think that probably isn't uncommon to have those kind of feelings. As always, I need to keep working on my wait time and allowing them time to think, because we know that with wait time the answers get longer, they get more detailed, they come up with things that they might not have thought of and we didn't expect them to come up with, that we can hitchhike on and move away from in allowing others to come into the discussion. So I think overall I was pleased. I would do things differently next time, but I think that's the nature of teaching. Whenever I have to think about structuring any lesson, the same holds true. I need to think about, what do I want them to know? Why do they need to know it? What are the key understandings I want them to come out with? So that no activity, no strategy overtakes what my outcomes are. Then I need to decide what's the best way of reaching that. And many times I like to bring in the inquiry piece of it, because it allows for deeper understandings. In the case of the lesson that we watched the students engaging in the protozoans, I could have just lectured. I could have given them all the characteristics of the protist kingdom. I could have given them examples. And in fact, we did go through a PowerPoint presentation, and it isn't in their text, so they were provided with some background. They would have memorized it dutifully for the test and promptly forgotten it. And when suddenly the euglena and the paramecium and the amoebas are delivered to their 4th grade classroom, they'd remember doing something with it and having fun, but have no depth of understanding. What the inquiry activity allowed me to do in this case was to give them multiple examples, to build a rich neural connection so that they could really have lots of hooks to move toward as they think about it, and feel very confident of the diversity and how they could bring it to students and have them looking at characteristics as well. Because honestly, we're not interested that 4th graders can identify protozoans. There's no viable purpose in that. But to understand the complexity of life, to understand even at the single cell organism how structure function varies, whether they're photosynthetic, whether they're heterotrophs, whether they're, obviously autotrophs, to look at those characteristics and say, wow, they're all different, even though they're even just single cells. I use the 5 E's, and so I plan around the 5 E's. And the 5 E's are in a linear format. They're a circular format. So the evaluate is in the center, and then the other E's are engage, explore, expand, and explain. And if you think about it as a web, constantly going in and out of that evaluate, that's formative evaluation. And I need to listen to the students to see if I need to re-engage them, if I need to stop and do some explaining, either whole group or small group, if they need to expand on it more, to understand it more, or if perhaps they just need to explain it themselves and I need to listen to them to be sure they understand it. And so it's a very fluid arrangement. It isn't teach, test, and hope for the best. It's a matter of listening to the talk back of the situation, as Sean would say. I can't tell them everything they need to know in the beginning because it'd be too soon. And if I wait too long, it'll be too late. So I really need to allow that fluid interchange occur within the classroom. There are those main concepts, those non-negotiables. I wanted them to, as I said, understand diversity, to understand characteristics of eukaryotes. I want them to understand the structure function. Those were non-negotiables. And as they worked through it, that just kept coming through in theme and variation. But I kind of like to think of myself as on roller skates, moving around and listening to them and interacting with them. And it's a fun way to teach. And I think develops a much deeper understanding than a superficial knowledge level. Formal assessments, I believe in multiple forms of assessment. The students certainly are assessed on the labs they turn in. There's a checklist that they're given for all their labs that they're evaluated against. They certainly get traditional tests. There are certain things they need to know. It is a biology course, after all, and there are certain background information that they need to have and to be able to explain. I like to do short answer essays a lot in that, so I can uncover those explanations more than multiple guess would. They have a project they do, and that has a checklist and a rubric that goes with it. I believe if you're going to teach in a more inquiry-based format, then you need to assess in a more performance-based format. But I also believe there's no fatal flaw when you balance assessments at 20%, 20%, 20%. They have multiple opportunities to demonstrate understanding in multiple ways. Reality is when they're at the college level, they've been presented those facts too many times. It's time that they begin to apply it, to understand it, and to explain it in more depth of how it fits together. And frankly, if they use those vocabulary words in the context, in their explanation, they're getting a much deeper understanding than a definition. So, as Wiggins and McTighe would say, backward design. What are those key outcomes I'm after? How am I going to know if they reach it? The assessment piece. What activities am I going to plan to help them get there? And then what's my toolbox? What am I going to pull from materials to make that happen? And it works well with inquiry. Inquiry is often thought about as a field of science because that's how scientists do in the real world, looking at investigations. There's a sort of misunderstanding that all scientists do the scientific method like the Ten Commandments that's in granite. When reality is most scientists tend to look at something and say, oh, wonder what would happen if we tried this. Do you think this same thing would be true in another case? And then set up hypotheses based on that. It's not an educated guess, as some people like to say. It's really based on some background observations. It looks different in each of the sciences. It looks different in physics as it's a more designed world engineering type focus as compared to chemistry and biology, where we do test hypotheses and collect data in a different way. but it works in lots of other content areas as well. The claims and evidence piece I used for the science writing heuristic could be just as easily used to defend an argument. You could do main idea and supporting details if you were looking at different types of written text, primary and secondary sources. You could certainly look at thesis proof kind of statements. There's lots of variations in that can be used in other content areas. The overarching idea is to engage the students deeply in the learning so they own it. That's what's critical. It's not necessarily, we can't hand them our understanding. It has to be something that engages them, that interests them, that excites them, that they want to learn more about. We do it already as we move them, for example, through their theology classes. And they finally get to that P course. And they need to finally say, here's my beliefs, and it's based on biblical principles, it's based on other theology classes, but we're moving them to maturity of their own construction of their understanding. We can't hand them our faith. It won't stand well at all. It will quickly falter if conditions are difficult. So they need to get that depth of understanding. We can't hand them our belief system in anything. We do it through capstone projects. We do it through senior seminars. In the sciences, we have independent science investigations or lit reviews. There's art expositions. Those are the kind of things where they have taken that ownership to their own level of understanding, of interest. We've done it on our dissertations. It's something we care about, we want to live with, and we continue to explore. If we can move them into that way of thinking, instead of the thinking of, what do you want me to do? Is this good enough? Is this what you wanted? But instead they own it. And that's where we're creating lifelong learners. For example, we could lecture and lecture on reconciliation. We can give them all the research. We can talk about different cultures. But if we immerse them in a culture, learning traditions, learning belief systems, learning history, and the issues facing the people, they're going to develop a much deeper understanding and a willingness to move to reconciliation and caring. It's capturing not just the head but the heart. They're not standing back like the two-way mirror observer, but they're immersed there. They're part of someone else's lives for a long period of time, and that changes their hearts. And that's what we want to look at in any learning. It's going to capture different students in different content areas because we all have different passions. We're looking at those big essential questions that stand over time, that are authentic. Certainly in the sciences, we look at homeostasis, we look at structure function, we look at transfer of energy. They're going to come up course after course, year after year, in different theme and variations. We look at big things like, why does conflict occur? And what does it mean to be free? It's not fuzzy things like change. What's that mean? But instead it's things that go to the heart of the discipline. It might be in the arts. You might talk about harmony and composition. In history, you might talk about perspectives and voices, migration, different things that will occur. The experts know what those big themes are that are going to continue through it. Could list them much more quickly than I can. But the idea is that we're not focusing on facts and definitions. Why are we teaching the concept? What do we want them to understand? And how are we going to get them there? And that's how we structure the inquiry. We're moving them from novices to experts. And an expert can describe various nuances of things. If you look at your hobbies or whatever, that you can describe in great detail all the little idiosyncrasies that nobody could give you a multiple choice test on. That's where we want to move them toward in an inquiry example. They should be able to give examples of it, non-examples. They should be able to explain it. They want to know more about it. Theme, variations, all those kind of things. They own it. It's in depth. They might look at primary, secondary sources. They might conduct interviews. They might undergo debates. They might do presentations. They might go testify before a legislative committee. It's testing that knowledge at a deeper level. Because in the end, it's not what we know. I mean, who cares? They're with us for a very short amount of time. We can't pour the information in their heads. Or as my grandfather used to always say, we can't put an old head on a young body. They have to develop that passion for learning and for understanding. And we can't do it by handing it to them. We have to empower them to actually want to learn more, to understand it for themselves. So it's not these black-white arguments you're hearing all the time. You know, does global climate change exist or not? It's not the silly little sound bites you hear in the media or the political debates over health care without any depth of understanding on either side. It's that taking something and turning it in different directions and looking at it through different lenses and through different angles and understanding nuances and how it affects you and how it affects me and how it affects someone else. And that's how we're going to move toward an actual literate populace instead of a fast-paced group of people going nowhere.